All right, it's 11 o'clock, so let's get started. Thank you for joining us today. My name is Carrie Phillips, and I'll be coordinating today's webinar. I'm here with Jackie Carville. In this webinar, Jackie will provide a sneak peek of the new features coming to MegAlign Pro in Lasergene 12. You may have noticed your phone has been muted. However, we encourage you to ask questions. To ask a question, type it into the chat dialog and send to host. I will then direct these questions to Jackie to be answered for the whole group. If you need any assistance or have any questions during the webinar, you can send a chat message to me using the chat box. And with that, I'll turn it over to Jackie.
Hi there, thanks Carrie. So to get started today, I'll be giving some background on DNASTAR as a company, as well as MegAlign Pro itself. And then I'll be getting into the software where I'll preview the new MegAlign Pro features that are coming soon in Lasergene 12. So as a little bit about us, we are a global software company headquartered here in Madison, Wisconsin. Our sales, marketing, developers, and support network are all located here. So if you have any questions or need support, we do have the full team available to answer you, and then relay your feedback to development to make our software even better in future releases. As some history about the company, we were born out of the E. coli lab of Dr. Fred Blattner, who was a professor of genetics at the University of Wisconsin in 1984. We built a strong reputation with our early products during the Sanger sequencing years that still continues on today. This year we're celebrating our 30th anniversary, so we have some materials on our website I'd encourage you to check out for that. While still offering some of these classic sequence assembly and analysis tools, we have expanded into the realms of next-gen sequencing, supporting all major next-gen sequencing platforms. In addition, we also now offer software to assist the work of structural biologists. Now, our products have long been established on the desktop computer, but are additionally available both on the Linux cluster or on the new DNASTAR cloud. As a note before we proceed with the webinar, our software is currently designed for research use only. Now, as we can see from this graph, DNASTAR is the leader in citations in peer-reviewed journals. For the past 30 years, more researchers have cited our software in scientific journals than any other commercial sequence analysis software available on the market today. Here at DNASTAR, we also offer 3 different suites or software packages for different areas of research. Lasergene 12, which is coming soon, offers significant enhancements to these different suites. Now, the Lasergene Genomics Suite provides all of the software necessary for next-gen sequence assembly and analysis. With the release of Lasergene 12, several new workflows in Lasergene Genomics Suite are going to be introduced, including enhanced SNP prediction accuracy as well as new gene panel workflows. I would encourage you to check out our website. We'll have a webinar later this month on those particular gene panel topics if that's something that interests you as well. The Structural Biology Suite provides tools for protein sequence and structure analysis. Our protein structure prediction program, NovaFold, also complements the Structural Biology Suite. And finally, we have our Evolution Suite, which we'll be focusing on for the rest of today's presentation. With the release of Lasergene 12, new sequence alignment methods and tools were incorporated into the MegAlign Pro application within the Evolution Suite. Finally, all of these suites can be purchased, as I mentioned earlier, on both the desktop computer or on the DNASTAR cloud. Now, as a little more on this Evolution Suite, this is our suite for evolutionary biologists to work with sequence alignments. This suite's designed around MegAlign Pro, which is the application we'll be working with today. Now, the applications in the Evolution Suite support a variety of different workflows, in addition to some of the multiple sequence alignments and profile alignments and phylogenetic analysis. So in addition to those, it also supports topics such as primer design, virtual cloning, gene discovery, Sanger sequence assembly, and such topics like that. So today we'll be looking at several new features which are in MegAlign Pro version 12. Some of these new features include the MAFFT alignment algorithm, as well as some new profile alignment functionality, which includes merging alignments together, as well as adding sequences and merging those new sequences into existing alignments. We also have some nice new sequence tracks that are available that we'll take a look at, as well as some enhanced tree building functionality. So our agenda for today, first we'll sit down and we'll take a look at the interface of the application. Second, we'll go through some different workflows that showcase some of these different alignment algorithms that we have available. Next, we'll look at some profile alignment functionality, and then we'll wrap up with a look at some phylogenetic tree analysis. I'm going to get out of the presentation here, and I'm going to go into the software. The first thing I want to show you here is our DNASTAR navigator. Open that up here. And the navigator, as this opens up, is you'll see is a guide to all of our DNASTAR applications. So we can see this here. You can view a summary of all the DNASTAR applications that are available to you here. And if we hover over any of these icons, such as MegAlign Pro that we'll be looking at today, we can see this nice little pop-up where we can read a more in-depth description of each application, as well as access additional resources such as videos, FAQs, tutorials, and demo data for that particular product. Another nice thing about the navigator is that it's fully integrated with our current help system. So you can use this to search across all of our products for answers to any questions you might have. I'm going to get out of the navigator and then we'll go straight into MegAlign Pro here. So this is our MegAlign Pro application. I've got a new blank project opened up here. So for this first workflow, what I plan on doing is introducing the various views in the MegAlign Pro interface while walking you through how to perform an easy multiple alignment in MegAlign Pro. So in this workflow, the plan is that we'll be adding some protein sequences into our MegAlign Pro project, and then we'll align those protein sequences using our newly added MAFFT algorithm. So the first thing we need to do, obviously, here is add in our sequences into the project. And we can do this in a couple of ways. So we can either select File, Add Sequences, or we can go up to the toolbar at the top of the screen here and select our Add Sequences to Project icon. So I'm going to go ahead and do that. And here, then I'm going to go ahead and I'm going to use these 5 alcohol dehydrogenase protein sequences. These are all from different animal species, so I've highlighted all of these sequences, and I'll go ahead and open them in the project. And once I have these sequences opened here in MegAlign Pro, we can take a look at some of the different windows we have available for use here. Now, the first view that we're looking at here is our overview panel, which is right here at the top of the screen. And the overview exists to provide a way to navigate within the sequences, no matter what type of sequences or alignment is used here. So prior to alignment, as you can see, we've got the unaligned sequences present here. Each of our imported sequences appears in the overview as a gray block with no gaps. And so we can see that all of these gray blocks have different lengths here, which is proportional to the ungapped length of the corresponding sequence. We can use this overview panel as a sequence navigation tool in a couple different ways. So the light blue region at the top here is called the viewport, and this shows the region currently being displayed within the sequences view on the bottom here. So we can slide this viewport manually to go to any desired region that we wish, or we can actually double-click on any residue, and that'll center the viewport around that particular region. So now to the right side of the screen, we can see the Style panel here. This Style panel we'll be looking at throughout the webinar today, but this offers us the option to customize parameters for any of the views that we're going to be looking at in MegAlign Pro. But let's go ahead for now and we'll align our 5 alcohol dehydrogenase sequences. So as I mentioned, for this workflow, we're going to be aligning using the MAFFT algorithm, as this is a brand new algorithm that we incorporated for Lasergene 12. But obviously you can certainly use some of the other alignment algorithms that we have available. So we can do this again in a couple ways. So we can do this by selecting Align using MAFFT, or as I said, any of the other algorithms that are located here. Or again, we can go to the toolbar And from the drop-down menu here, we can also click Align using MAFFT. So I'm going to go ahead and do that here. This will just take a couple seconds for this alignment to complete, and there we go. So now if we look at the overview, we notice now that all of these sequence blocks are the same length, and they've also turned green, so we know that these have been aligned here. We can also see the sequence gaps available in these blocks, which are represented by the gray coloring. So now that we've performed this alignment, we can actually explore the sequences view a little more closely. So sequences view is located right here at the bottom of the screen here. And any of the sequences selected in this view are simultaneously selected in all other MegAlign Pro views. So you can see if I click on the sequence here, that's also going to be highlighted up here in the overview panel. At the top of the sequences view, we can see the consensus sequence and consensus ruler up at the top. And then in the sequences area here on the side, we can see the names and codes for each of our sequences. Now the names of each of these sequences that we can see here originated from the locus names which were embedded in our input sequence files. And so we want to be able to change these names so we can more easily perform analysis on our alignment. So to do this is really simple. I can show you how to rename this. So we just right-click on the sequence names and any of the views to select it and choose Rename. And then I can go ahead and rename these as desired. So I can go through and rename based on the different animal species that are present in the various samples. All right, so now we can more easily see what we're working with and see that that has been simultaneously updated, not only just in the sequences view here, but up in the overview as well. So for each of these sequences, we can choose to expand the view by selecting the plus icon. And again, we can do that in all of the views here. So right now we're looking at some features. And in this details track, as well as a gap-aware ruler. We'll be looking at some more of these track functionality later, but I just wanted to point that out briefly at this point in the webinar. Again, if we wish to change the appearance of the sequences themselves, we can look to the sequences bar within the style panel. So we can see all of these different nice options we have available for styling our sequences view. So here we have the option to change the font and font size. We can also change the layout if we want to include some wrapping. That's nice for if you want to print out the sequences view later. Additionally, with Lasergene 12, we've also included some new coloring options. So previous versions of MegAlign Pro, we've always had these few different coloring templates here that you can choose to use. But now we also have some comparison abilities where we can use coloring methods to compare the different sequences in the sequences view. So if I want to color only differences from reference, I can do that, and I can choose a solid color to show that as well. So again, that's just a nice option. Lots of different customization options available to you here within the style panel, just so you can use these when you're customizing your own publication quality graphics here. So again, finally, the sequences view, as we're looking at here, it also features some sequence logos, which we can see on the bottom of the screen. These sequence logos are brand new, again, for Lasergene 12 here. The sequence logo, if you're not familiar, is a visualization of the nucleotide or amino acid at a given position. The height ranges from 0 to 2 for nucleotides, or from 0 to 4.3 for protein sequences that we see here. This is proportional to the amount of information which is available for each character at that position. So if we hover over any of these sequence logos here, if I hover over this position, we can reveal a tooltip with the information content of each letter at that particular position. So now I'm going to go ahead and click on the Distance tab here at the bottom of the MegAlign Pro window, and we're going to take a look at the distance view here. Now, the distance view shows a matrix of numbers representing distances between each pair of sequences. Now, by default, the distance view matrix is calculated using uncorrected pairwise distance. However, again, if we go to the Style panel and we expand up our distance view, we can see that we have some different parameters and options available here. So from this default uncorrected pairwise distance, we can also go ahead and select some of these different metrics here. And if you watch the distance table here, if I select on that, we can see that the numbers do change to coordinate with that new metric. So another option to point out is that the upper and lower triangles of the distance matrix are reflections of each other. So they will contain the same information, just something to keep in mind there. And again, finally, as I showed you, the values differ for each of the various metrics used. So there are many useful resources located in the DNASTAR help for assistance interpreting the information for each specific metric that you choose. So you can now access help for each application, either through the application itself or through our website. Again, just to mention, from the DNASTAR homepage, if you go under the Support tab, you can select Online Help and access it for any of these applications. So a last point about this distance metrics is that we can easily export this. So we can do File, Export Data here, and then select the distance matrix to export that particular file for further use. So that wraps up our first workflow here, looking at just a simple multi-sample alignment here, or multiple sequence alignment. And so I'm going to go ahead and open up a new alignment project for the next work that we're going to do here. So for this next particular example here, we're going to look at how to work with multi-segment files in a project. So sequences that consist of more than one chromosome, contig, or fragment are called multi-segment files. So let's say that you want to string together the consensus sequence from all the contigs in an alignment, or all the chromosomes in a genome, or all the exons in a gene. If you simply added these sequences as we did in the previous workflow, you're going to end up aligning these parts to one another. Instead, we need to enter in the sequences using a different method to make sure the entire set of sequences in the multi-segment file will be treated as an end-to-end concatenation instead of aligning all of these parts to each other. So instead of simply clicking this Add Sequences to Project button here, what we're going to want to look at is the Add Sequences End-to-End in Project option. Again, this option can be accessed from the toolbar as well as under the File menu here. So once I select that, it brings up this nice little window here for adding sequences end-to-end. So we're going to click Add Button, and we're going to navigate to our specific data that we're going to use. So here we're going to be using nucleotide files that represent the RP1 exons for 7 mammal species. Now, each exon here consists of 3 to 4 different segments. We're going to want to sort our files to make sure that they're all not only alphabetical, but also in numerical order here. So the first one we're going to do, I'll just select the 4 chimp segments and open those up. I'm going to change the default name. And I'm going to want to make sure that this Add Another box is checked in this window, because we're going to add in a bunch of these animals here. So we're going to click OK. And we can see that that comes up behind the screen. And we can watch as we quickly go through and add these different files here. Again, we're going to use many different animal species to give us a look at some of the interesting gapping and such that goes on here. So add in this mouse exon. In the rat. Now that we're down to our last group, we're going to take a look at, we're going to purposefully add this group in incorrectly. So we can take a look at how adding some of these groups out of order can impact your final alignment. We'll see what happens there. So I'm going to open up these rhesus exons here, rename the file, and then I'm going to order them, I think, let's do 4, 2, 3, 1 instead of 1, 2, 3, 4. There we go. We're not going to want to add any other ones after this, and we're going to click OK. And so by doing this, initial adding before we even align these sequences, I'm going to go ahead and expand down so we can take a look at the overview up here. And we can already kind of see that this rhesus group looks pretty different from the other ones, just being out of order there. But now that we've created all of our groups, I'm going to go ahead and show you the final alignment for this option. Now, since we have a lot of different groups in this multi-segment file, this will take maybe 3 minutes to align here. But so we don't have to wait around for that, I'll go ahead and open that up in the project. So this is the alignment. I performed this with the muscle alignment algorithm, and this is the one where we aligned it incorrectly with that last rhesus group out of order here. And so the first thing that we're going to want to do is we're going to look for the longest segment as well as the longest gap region in each sequence. And this is usually a pretty good idea to just look at after you've performed your alignment to make sure that, to get that first initial idea whether this is aligning correctly. So what we can see here for all of these sequences, we can see that this longest segment is here on the right, and at the furthermost right side we have this gappy, large gappy section. However, for the rhesus example, where we arrange this in a different order, we can see that this longest segment is on the left as well as that longest gapped region. So the nice thing is that in MegAlign Pro, it's really easy to remove a group and then add it again in the proper order. So all we have to do is right-click on the rhesus group and select Remove Sequence. And then we can really easily go ahead and add in another sequence, add it back into the project, do another end-to-end add, and then add that sequence in the right order. So let me quickly open up. I've again performed this alignment using muscle with these aligned correctly. I'm going to expand this down. And then now if we look at this rhesus group here, we can see that now we have a pretty good idea that these are, again, located in the proper order. And we can also note that the largest gap region in the overview has kind of shortened here. So now with this alignment, we have some of these shorter gap regions, and it's less gappy because we've aligned this correctly. So that wraps up how to work on multi-segment file alignments. So one other view I'd like to mention as we move forward is our new to Lasergene 12 tracks panel. So I'm going to open this up. This is located next to the style panel on the right part of the screen here. So we can open up the tracks panel and we can see different track options available here on the side. These we can all see best. We can see them in any view, but I'd like to look at them in the sequences view today, so I'm going to expand that up for us. We'll maybe scroll through this alignment a little bit to give us a better idea. So as we look in this panel, depending on whether you are using DNA, RNA, or protein sequences, your track options are going to be different. These are really easy to look at. You can simply check and uncheck the different boxes to choose what you would like to view or what you would like to hide. So we're going to look at some of these tracks with our current alignment here. So let me just expand out. Let's look at these dog sequences here. And so again, as we can see under the sequence details tracks here, we have both the features and the ruler checked, which we can see checked under this sequence. We can also look at translation as I check that. In the translation view here, we can look at that particular track. Stop codons are represented by dots that we see here. We can also go ahead and look at GC content in this area. This graph shows the GC content on a vertical scale of 0 to 1. And similarly, the gap fraction, we might have to scroll a little bit to get to that there. Gap fraction is also shown on a vertical scale of 0 to 1 as well. While we're looking at some of the tracks here, another nice function we have is the ability to copy any of the features to the consensus sequence that we're using. So I'm right-clicking on this particular feature here. I can select to copy that. And then if I expand down the consensus sequence here, we can see that we've successfully copied this feature up. And again, we can do this with any of our features that are found here and add those quickly and easily up to that consensus sequence. So one last interesting view that I'd like to note as we move through this is our console view. And so we can go ahead and open that up by selecting View, Console. And this opens up a log that shows the progress of your alignment. And for an alignment in progress, the console displays the start time and the progress information. For a completed alignment, just like this one, the console displays statistics for the alignment as well as a start and end time. And again, this can be pretty helpful if you do run into any issues with MegAlign Pro. Simply export this console text, and that could be helpful to our technical support team to find out what's going on within your particular project. So this wraps up our workflow for handling multi-segment files. So now I'm going to move on from that, and I'll demonstrate how to perform a whole genome alignment. So for this particular tutorial, the plan is we're going to add 6 different nucleotide sequences into MegAlign Pro, and then we're going to go ahead and align them using the progressive MAFFT algorithm. So I'm going to clean up the screen a little bit here, take out some of these projects. So I'm going to go ahead and open up. I've already performed this MAFFT alignment here. MAFFT alignments usually take, for this one I had several bacterial genomes, maybe 5 to 10 minutes. So I just want to make sure to have this open and up for us to be ready to look at right away. So as a little bit of information about the MAFFT algorithm, this algorithm is specifically designed to align genomes, which is a workflow that's usually impossible or at least very difficult with other alignment methods. There are a few characteristics of these whole genome alignments that can cause problems in some of these other methods. Now first, these whole genomes have a large memory requirement. Second, genomes can be very divergent. Genomes can also have lots of rearrangements, including both large and small insertions, deletions, duplications, and inversions. Some genomes may also have gene clusters or large islands of unique sequences. And finally, you may also run into some problems with transposition in eukaryotes, where entire sections of chromosomes in one genome may be transposed to a different chromosome in other genomes. Now, the MAFFT alignment can, to some extent, surmount some of these issues. How this method works is to break the problem into smaller alignments, and at the end, merge as many of these small alignments together as possible. So again, as I said, I've been working with some H. pylori bacterial genomes to create this alignment here. And these, can be added into our project just as we've done with some of the other workflows here, or just by dragging and dropping them into the overview panel. For most alignment methods, too, I want to point out, even though this alignment's already been completed, that you can align with any of these algorithms and adjust the different parameters and options. So all I did here is I selected from this drop-down menu here, Align with Options. This is also available under the Align tab as well. And so we can look at here in this Align Sequences view, that we have some of the many different parameters that we can choose to adjust for any of these alignment methods here. And what I did for this particular MAFFT alignment, and I'll show you, is I changed the default seed weight, which is a good option here in MAFFT. In MAFFT, the seed weight is the minimum weight of the seed pattern used during the first pass of an alignment. When aligning divergent genomes, lower seed weights may provide better sensitivity. However, if the value is set too low, the result will be reduced sensitivity. So for this workflow, what I did was I just unchecked the default seed weight and I lowered this seed weight to 13 here instead of the default 15. So again, if I had just loaded in the sequences, I could just click Align and go ahead with that, but I've already performed this alignment, so we can jump right into that and look at the completed project today. So after doing a genomic sequence alignment with MAFFT, MegAlign Pro does display the genomes quite differently than some of the other alignments that we've already looked at in the webinar today. So how they display this alignment is as locally collinear blocks. Now these blocks represent multiply aligned sections of at least 2 of the current set of genomic sequences. Discontinuity in numbering is displayed as a break between the different blocks, and the connecting lines that we see here show the correspondence between the different blocks. So if we notice some of the blocks hanging below the black line, as we can see, for example, in this blue block here, this indicates that the hanging block is on the opposite strand as compared to the other blocks. So we can see here that all of these blocks are of different colors. If I double click on a block, let's say I click on this gold block here, we can see now that all the gold blocks are now in a single blue highlighted column. MegAlign Pro will flip strands if necessary to orient properly for the new alignment. And then the viewport is now located, as we can see, at the leftmost end of these blocks. Now if we look down in the sequences view below, we can see that the residues outside the selected box here are shown in a dimmed version of the color scheme. And these here represent flanking sequences that are immediately adjacent to the currently selected block and appear as unaligned, ungapped, and without a consensus. If we want to see additional details for these dimmed sequences, we have to select the corresponding block in the overview. But what is bright and in our color scheme is what the viewport is currently centered about in the selected block, and so we can easily scroll through our selected block to using this viewport, as we've done in some of the other alignments here. That wraps up my overview on the multiple genome alignments here. I can take a break now for any questions if we have any.
Sure, Jackie, we do just have one question. What file types can be imported into MegAlign Pro?
Sure. If I select this Add Sequences to Project option here, I can kind of show you the different formats that are available. So as we can see, we use a lot of different file formats outside of just our normal, some of our own Lasergene files here. So we can also use FASTA files, GenBank files, different DNASTAR Lasergene files that you might have from some of our other applications like .seq or .pro files or .mseq files. SeqNinja as well is another Lasergene application. And again, other Lasergene documents here. But we'll look at a little later too. We can load in completed alignments, these .msa files. So lots of different known formats that you can choose to import to use in your different projects. All right. So now what I'd like to move on to and switch gears a little bit, since we've looked at some of these basic alignments in MegAlign Pro, I'd like to look at some profile alignments, as these are new functionality here in Lasergene 12. So with this functionality comes the ability to add additional sequences to an existing alignment, as well as merging 2 alignments together. So let me just open up a brand new project here for us to work with. And the first thing we're going to do here is take a look at how we merge additional unaligned sequences to an existing alignment that we already have. So let's go back to using our alcohol dehydrogenase sequences that we used in the first example here. So this time I'll go ahead and load in some of these sequences from some of my sample data here. I'll load in 3 of these sequences instead of all 5 and select Open. And we can see those 3 sequences come up in our views. So I think what I'll do here is just quickly perform a MAFFT alignment and get those 3 sequences aligned together. And then I'm going to go ahead and add in the 2 remaining sequences that I didn't add. And so this kind of starts some of the new functionality here. So now we can see, instead of just having one one overview window and one sequences view, we have 2 of each. And these are all titled. So on the top here we have the aligned sequences, or the aligned sequences overview, as well as the unaligned sequences overview, and the same with the bottom here. So this just lets us take a quick look at, what we're working with, and from these different coloring options, we can keep these straight, which ones are aligned and which ones aren't. From doing this, we can also see that now we have a new icon available up in the toolbar, which is the Merge Unaligned Sequences into this document's existing alignment view. So I'm going to go ahead and select this option. And then this brings up some parameters that we can adjust. So again, we can, merge these in using a different alignment algorithm and adjust some other options as well. But I'll go ahead and just leave these at their defaults and click Align. And now we can see after this alignment is complete, we've gone back down to just the two different views here, and we can see that our unaligned sequences have been merged into the existing alignment document. Now we're going to take a look at a slightly different example. And for this particular case, we'll look at how to perform a novel alignment of formerly aligned sequences and selected unaligned sequences. So for this case, you may have already aligned sequences, but then you decide to realign the same sequences plus just some selected unaligned ones. So again, I'll just add in, I'll just do 2 of these right here. And then I'll add in a few, and we'll do a quick MAFFT alignment. And now I'll go ahead and add in the remaining 3 to get that nice 2-window view again. So let's say I have these 3 sequences loaded in here, and I just want to merge in the singular one here. I'll select that. And now I'll go up to the Align menu here, and I'll select Align, Realign with Selected Unaligned. Again, we'll get this nice parameters window that comes up. Click Align. And again, now we can see if I open up this overview panel a little more, you can see that that 3rd alignment has merged in together, and we've realigned all 3 of these sequences. And the final profile alignment functionality new for Lasergene 12 here is that I'll discuss how to merge 2 different alignments together. I'm going to open a file here. And so I've got a couple different ADH alignments already done. And one I've done with some human muscle and another with some mouse muscle samples here. So I'll open up the human one. So we have all of these different sequences here that have already been aligned in this particular alignment here. So I've loaded in this .msa file, and I've seen the alignment come up in our MegAlign Pro views. And now to add the mouse alignment, I'm going to select Align, Merge Alignment into Existing Alignment. And then I'll navigate and select in the mouse and click Open. And again, we get this nice parameters window. We can adjust any of the parameters we want, but I'll just go ahead and click Align. And now we can see very, very quickly here we've merged these 2 alignments together. Now I have all the different sequences from both of these alignments displayed, and we can go ahead about our analysis as we wish. So finally, the last thing I'd like to talk about in this webinar today are some of our capabilities to create different phylogenetic trees. So after performing an alignment, you can access the MegAlign Pro tree view from this bottom panel here, similarly to how we did the distances view. We also have some styling options right here in MegAlign Pro to go ahead and adjust this tree. Again, we have some nice customization options. We can do the font size. We can color the branches in different ways if we wish. And another nice new styling option we have here in Lasergene 12 is showing the branch length right within the figure here. We can also adjust the decimal places for this particular branch length as well if desired. But again, new to Lasergene 12, another nice functionality that we have here is that we can integrate our trees here and our alignments with some external tree viewers such as Figtree. So I've already gone ahead and downloaded Figtree onto this computer. We have a link to Figtree on our website where you can download it from there as well. But what I can easily do here is select File, Send Tree to Figtree. And really quickly here, it's kind of that one-click export there. We've opened up the Figtree application with our existing tree from MegAlign Pro, and we can go ahead and do some different styling options here in Figtree as well. There's lots of nice different options in here that you can go ahead and do to create some of these publication quality graphics for you. So that wraps up my demonstration within the software today. Before we wrap up, I'd like to point out a few different support options that are aside from our webinars that are available as you're working your way through MegAlign Pro. So I'll go out of here and go pull up our website here. So if we go to our website, we have this support tab up at the top here. And as you can see under the support tab, there's a variety of options available for you, such as online help, tutorials. One of the nice ones is our newly revamped video library. So this allows you to search. We've got over 200 videos in this video library and a variety of different workflows. We can also look at these videos by topics, so multiple and pairwise alignments that we worked with today, or by products, so I can go ahead and look at some of these MegAlign Pro and MegAlign videos that we have here. So these are pretty helpful videos that 2 to 3 minutes long and quickly walk you through some of our functionality. So that's about all from me today. I can take any other questions at this time from people.
Sure, Jackie. Thank you so much. We did have one question. The person writes, I've used DNASTAR's MegAlign in the past. What's the difference between MegAlign and MegAlign Pro?
Sure. So our MegAlign application is kind of one of our very classic applications from earlier in DNASTAR. There are still a couple pieces of functionality that you can find in MegAlign. I know those include bootstrapping as well as some pairwise alignment algorithms. I know there's a Jotunheim and things along those lines. So you can explore our MegAlign Pro page on our website to get a complete list, but essentially the differences are just that bootstrapping capability as well as a few of those other kind of classic pairwise alignment methods that you can still do. Again, there's also a nice, if you perform an alignment in MegAlign Pro and then you wish to do some of that analysis, you don't have to realign your sequences in the MegAlign Pro or in the classic MegAlign application. You can just simply export that directly to MegAlign.
Well, great. Well, thank you so much, Jackie. It looks like we're just about out of time. Some of the questions that I did receive during the webinar were too technical to address today. If I wasn't able to get to your question, we will follow up with you by email. And, of course, if you think of any other questions later, you can always email those to Jackie using webinars@dnastar.com or tweet @DNAStarInc on Twitter. And if you have any ideas for future webinar topics, please don't hesitate to send those to us. Thank you again for joining us today, and we hope you found this webinar helpful. Have a great day.